presentation mode? Oh,
Yes.
okay. I'm Luca Bargelloni from University of Padova. I'm a partner, University of Padova is partner of AQUA-FAANG, a large European project dedicated to functional annotation of the most important six European species for aquaculture. I will present you, as Shagla was saying, the case of the European Seabass. But to start, just a little introduction. I've stolen the title of Anti's first presentation in the first meeting of first webinar of this series. It was talking about the black box, because in fact, in most cases, genomics has been applied completely blindly or almost completely blindly in quantitative genetics applied to aquaculture. So I will try to convince you that it's now we have to dare and try to open the black box. It's not an easy task, but it's something that in human genomics and also in cattle and pig genomics is already ongoing. I think we are on the right track also for fish species. The black box is essentially this. We use SNPs, single nucleotide variants, in a blind way. We don't know what they are, what they the function they have, and so on. But in fact, in most cases, you have very few causative SNPs, either in the coding part of the gene or in the regulatory sequences, and many linked non-relevant markers linked to the causative ones. And especially for model species, it is now clear that most causative SNPs for most quantitative traits are located in regulatory sequence, which are non-coding, and therefore are very different, very difficult to identify and to understand their way of working. While now we have very nice softwares that can predict the effect of SNP encoding regions on the protein. So the challenge is to find these regulatory sequences, which are not only upstream of a specific gene. They can be very distant for the key gene for the trait, and also to understand which are the causative SNPs within regulatory region. Sorry. Okay. In fact, these regulatory regions control where a certain gene is expressed, either the tissue or the cells, specific cells. So it's very important because, as I said, most traits now are thought to be controlled by the expression of specific sets of genes. Also, they control whether a certain gene responds to specific stimuli. Like in this case, for instance, there are several immune genes that respond to the infection here with tilapia virus. You see in the black bars, you are upregulated the different genes, and they are in the control, they are downregulated. This is very important because, as I say, in most cases, these are the intermediate phenotypes that actually play a very big role in the macroscopic, in the classical traits, like in this case, resistance or immune response. How to find them? How to find those elusive regulatory regions? There is a general rule in eukaryote and invertebrate genomes that if a gene is not working, is repressed, the chromatin, which is a structure with DNA, the genomic DNA plus a certain set of proteins, mainly instons, is closed, so it's not accessible. Then when a gene is ready to be activated, but is not activated yet, it opens up the chromatin, it becomes permissive, and then you have the fully active gene when regulatory regions upstream or downstream or within the same gene are actually open, and the chromatin is modified, signaling that this is an active region, and this is actually tissue and cell specific and also condition specific. So we cannot find regulatory regions for in the entire genome just with one sample. We need to have a very big representation of different cells, different tissues, and different conditions. This is a big challenge, of course. So this is what we did. We started to do in AQUA-FAANG, especially in Work Package 3, which we call immune maps. It's and this I'm presenting this exactly because we are going to discuss about immune response and disease resistance. So 6 species, as I was mentioning before. We have the most important ones in Europe. You see here. We decided to work on different cell types. One was isolated leukocytes in vitro. You know, the leukocytes are the main immune cells in vertebrates, and also working on the all organ or head kidney. And for each of these species and each of these 2 types of cells or tissues, we use 3 different immune stimuli. One was, of course, the control, no stimulus, like a neutral buffer. Then we use poly-IC, which is synthetic double-strand RNA, which is sort of simulate a viral infection. And also we had a species of Vibrio, but inactivated, so simulating a bacterial infection. And for all these different samples and conditions, we used 4 to 6 biological replicates, so different individuals or cells stimulated in a different way. And we used 3 different levels of analysis. One was a classical analysis, now RNA-seq, which measure, as was showing before, the level of expression of a certain gene. Then we use another technology, maybe it's a bit less common or less familiar with you, which is called ATAC-seq, which actually is able to find accessible regions in the chromatin. And finally, another technology, which is called ChIP-seq. Don't bother about the acronyms, clearly a lot of technical jargon, but what is important is that they give you, this technology gives you the status, is the chromatin permissive, repressed, or active in a very variegated way. Okay, and this is the results of this kind of annotation in a specific region of the sea. Okay, so I was explaining that this is a specific region in the seabass genome where actually we have different genes which are expressed here at the RNA level, but not in all conditions. You see, when you stimulate with poly-IC, you have expression of this, and to less extent this gene, but not of this. And when you have the PBS, so the control buffer, you have almost no expression apart from this gene. In the color bar, which is very colorful, you have actually the chromatin status. You see here this gene, especially in this region here in red, and this region here in red, are very active chromatin regions under the stimuli of a simulated viral infection, and they are almost not active when you stimulate the cells with the control buffer. This is not a region, a random region. Is actually a region where we end up using a classical approach of quantitative genetics. We started with across 25 males and 25 females European seabass, and we produced an experimental F1 offspring where we had over 1,000 fish infected with the nodavirus, the infectious, the agent of nervous necrosis, viral nervous necrosis. You see here, and we surveyed the survival for 3 weeks, and we recorded the survival. And another group of fish were kept separate and were infected or not. We did not wait for them to die. We stopped them at 48 hours, and we were either control or infected, mock infected or controls. And then we analyzed the global transcriptome of this fish. And from those 2 different phenotypes, one phenotype was the classical survival data, and the other one was actually the RNA level expression of all the coding genes in the genome of the European seabass. We had we coupled that with the genotype data, which were actually produced with the SNP array, but then we imputed the whole genome sequence for each of the individuals. We did the classical GWAS for survival, associating with different genotypes. But the same with another phenotype here, which is the gene expression, and we have here, in fact, the EQTLs. So genotype variants which are associated not with the survival, but with the expression of individual genes. Plus, we applied this ATAC-seq with the accessibility of genome of the genome in the chromatin in the genome for both 10 controls and 10 infected fish. And what we found for GWAS, both for the phenotype of survival and the phenotype for the expression, is that one region was very significant in linkage group 12, explaining almost 40% of the genetic variance of survival, and also very much correlating with the expression of at least one, possibly two genes in the same QTL regions where we had the genotype for the QTL for survival. So the next step is now, we are currently going, we put everything together. So the survival SNPs linked to survival, the SNPs linked to gene expression, specific genes in the region, the profile of accessibility, you see the peaks identifying the region which are control regions, regulatory regions in the QTL area, in the QTL part of the genome. And then what we are doing now, analyzing these open chromatin regions where the SNPs, the candidate SNPs are, to see if they have effect on transcription factor, which are the protein regulating the expression or repressing the expression of genes. So ideally, this integrated approach should produce this kind of putative causative SNPs in this region. So to sum up, we think that this is a nice example of how integrating different information, we can actually find out the regulatory regions and possibly pinpointing the non-coding causative SNPs, which are very difficult, very elusive to identify. So 35 to 30% of genetic variance, as I said, single QTL. And when we go there, we have a lot of linked SNPs, but using this kind of integrated evidence, we might end up with just one or possibly two single causative SNPs, which would be much interesting and powerful for marker-assisted selection or even genomic selection. And I think I can stop here. Sorry for the interruption.